Welcome back. We are in our third method of determining physical interactions between two or more proteins in cell biology. The third method is generically, and there's a bunch of different ways to do it, but generically it's called a pull down assay. In other words, pull down, that's pulling out of solution. Okay, and there's a connection with affinity chromatography. So if you'll go back and look at your affinity chromatography notes to kind of review that. And so it's a method not inside the cell, but outside the cell. So it's an in vitro method. So remember, yeast 2 hybrid is inside the cell. Co-immunoprecipitation is outside the cell in vitro. And then now the pull down assay is in vitro. And again, this generic assay is looking for interaction protein partners. So it can confirm the existence of protein-protein interactions. And the verbiage there says predicted by other techniques, co-immunoprecipitation, yeast 2 hybrid, you could put that in your notes. And it's an additional screening for unknown protein interactions. In other words, we want to confirm what we think we know, right? Bioinformatics, etc., is looking at domains and making predictions. But what about protein-protein interactions that are through areas, domains that are unknown? Okay, so it's an investigation tool. Pull down assays are a form of affinity purification or affinity chromatography. Very much like immunoprecipitation, except now we're using the protein directly. We're not using an antibody to the protein. Okay, that was in the co-immunoprecipitation. It's also known as affinity purification, and the time and efficiency is higher than co-immunoprecipitation and probably a little bit more direct and a little bit more conclusive than yeast 2 hybrid. So it will give us a platform to perform the pull down or co-purification of partners, and we can change the environment, we can change the partners and see what differences. And this is some material from Thermo Fisher Life Sciences. So in a pull down assay, the bait protein, so we're not using an antibody, we're using the protein, is tagged and captured by an affinity ligand specific for the tag. The tag can be amino acids, it could be a chemical, a compound, a molecule, another protein. And so you generate a secondary affinity support for purifying other proteins that interact with it. So you usually immobilize the bait protein with its tag, and then you incubate that immobilized bait. That could be on beads, resin, or some type of plastic or a blot platform. And you pan through prey proteins such as a cell lysate. And remember our purification, we can break the cell into different fractions. Okay. Remember what happens sometimes if you just blend the whole cell up, you end up with proteins that are non-specific that seem to bind, but in reality, they're not ever in the same place. Okay, so kind of keep that in the back of your mind in discussion of this data, of these methods here. So a lot of times you are confirming what you suspect is a protein-protein interaction, or you're identifying an unknown. And again, it could be domain specific. It doesn't have to be the full protein. Okay. And then how you break apart and isolate to then do, say, 2D and then spot analysis through the multi-TOF or other methods, Edmond degradation, depends on uh, the nature of the tag and the binding here. So here's some, these are also known fusion tags, affinity tags, and their affinity binding ligands. So if we attach an enzyme, glutathione sulfur or S transferase. So this enzyme wants to transfer a sulfur molecule uh, onto its substrate, glutathione. So its affinity is glutathione. This enzyme binds to it, and it's very tight binding. There's something called polyhistidine, also known as polyhis, and these are usually 6 histidine proteins tagged on the end of any protein that you're calling bait. So, and then the histidine has a positive charge, and it will bind to nickel or cobalt complexes on an affinity column or beads. Okay, affinity or cobalt. So these molecules will capture all his tags. And there's a molecule known as biotin that binds to strepavidin. And by the way, this is the strongest protein-protein binding that's ever been detected, these two. So this is super strong here. If it's super strong, it's hard to break apart. So you're saying, well, do people use GST or the HIS? A lot of times it depends on what people are doing in the lab, in the school, in the department, uh, what may you've done previously. Okay, I'm not sure they're right in the beginning, you know which one's best. You might, under empirical studies, determine the uh, which one is better. If you did the pull down and got uh, poor reaction, then you might switch. Okay, see if there's something going on. So overall, when we're trying to figure out a gene function, we need to figure out what the protein does coded by that gene. And we've talked about protein-protein networks, and these interactions are, and they'll call them protein machinery here, protein machines. I got you some color here, which will assemble and disassemble, getting signals from inside, outside, from other cells, external cues. Everything is changing. Constantly, these machines are building, coming apart, assembling, disassembling. Things like cancer disease occurs when these machines don't assemble, or if they assemble, they don't disassemble, right? They keep going. So what proteins interact with what other proteins? You're looking for these pathway concepts here, pathways. And the GST is a very invaluable tool for us. This is kind of the first one, a little bit older than the HIS tag and biotin tag. And it's just something that is a little cheaper, I believe, than the HIS and the biotin. So here is a cartoon showing the GST pull down assay, and we'll go through this. This is some type of, this little pretzel stick here is representing glutathione affinity resin. So some type of inert molecule that they've attached glutathione. Also, is a protein of interest, our bait protein, which is the blue color here. So I guess I need to get something of a different color than blue here. Here it is. And this blue protein also has a GST molecule bound to it. So when you put that bait protein with the GST tag, it sticks because this enzyme is binding to glutathione. And so now this immobilizes the bait protein, and you've got the GST on one end, and you hope the areas for protein partner binding are exposed. And you say, how do you know the tag is not interfering? Well, you don't. That's why you do multiple different types and ways to look at it, okay? Because there may be other regions you can attach the tag and redo the experiment. Anyway, you allow this to occur, so the is attached to the glutathione resin. You add cell lysate, which has all these different types of cells, all these different colored shapes, and under the right pH, salt, other kinds of conditions, binding is thought to occur. So in this cartoon, 3 proteins bind to our bait protein. You then wash away the non-specific protein. Okay, you wash that away, and what you're left with is the proteins that bind to the bait. And then you elute, and this is empirical. This elution step here depends on the nature of the interactions. I've used high salt. I've used low pH. Either way, whatever you add to break apart the resin, you have to kind of neutralize it. If you use salt, you have to get rid of the salt. If you use acidic pH, you need to neutralize it. And at that point, what is pulled down, see, this is the idea, this pulled this complex down. We're then going back, and you can hit your notes to our SDS page analysis, 2D probably, and then the 2D spots, and we're using Maltitol Edmond or Maltitol fingerprinting, charge to mass. We're trying to identify these prey proteins, okay? And of course, once you get the prey proteins, you can actually attach them, say this purple diamond here, and do this same thing. And you would predict if you attach the purple diamond, assuming you're not doing to the area where they bind, you should be able to pull down the bait protein. Okay. Realize that the cell lysate is the variable here. This could be disease cells. This could be wild type. These could be mutant cells. These could be drug-treated cells, stress cells. So you can do a lot of variation here. And because the variation could be changing the expression level of the prey proteins. So you could do cancer and find the bait doesn't bind any of them. That would be very interesting. Is that the reason we have cancer? Is it because this machinery never builds? And could be the vice versa, that in wild type cells there's no binding, but in cancer these partners are bound, and they shouldn't be bound. So it's you keep track of this. Certainly the informatics keeps track of these data, and eventually, you know, we're to personalized medicine. How does your bait protein work in your system? Okay. You know, we'll try to infer that by your genomics, but, you know, if it's really important, they may even do a test. So you go to the doctor and get a GST pull down. Here's another cartoon, and it just shows the resin, or in this case, agarose beads. These are inert, and they kind of like an M&M, they coat it with glutathione substrate. And here is your tagged bait protein with GST. You mix, incubate the right temperature buffer. Here they are bounding together. Then you add the various lysates, and here's protein Y. And in this cartoon, we show the controls. Okay, it's very important to have a control here. Okay, this is control on this side. This is the bait on the other. So we have bait and GST. Here's glutathione cephalose beads. Okay, we add our lysate into this device. We apply centrifugal force. We get the liquid through. Okay, there's a little pore there, and the liquid comes through. The proteins that are going to bind to the bait attach. Then we elute them, and here's a 1D gel. And so we know this protein, but now we have protein 1, protein 2, protein. So we have 3 mystery proteins that bind. Notice that the green protein binds to GST. It doesn't bind to the bait. And you're saying, oh, we have 3 proteins. Well, the control does this for you. You just add GST by itself. You do the same reactions, and in this cartoon, look what binds to GST, the green protein. So when you run the control lane of what came down, you find there's GST. You're expecting that, and here is this protein, and you're saying, oh, this is non-specific binding. But notice the purple and the red proteins are not here. So you're implying they bind to bait. If you didn't have the control, you would spend 6 months working on this green protein. A simple control shows you that's irrelevant. You're saying, how can that work? Sometimes protein by chance will bind to other proteins, and they're not specific. And this is a way to get around that, running the proper controls here. So here's a cartoon from some company's website. It shows the in vitro prey synthesis kit, and the little P, orange P, are the prey. And then we add our bait protein or our GST with no bait, and we mix what we did, our prey. So this, in the previous experiment, okay, we did in vivo cell lysate. Now we're actually taking the gene for a predicted protein and making the protein in a test tube and then adding it and look for binding. And so this is when you have strong evidence, maybe to hybrid co-immunoprecipitation, and you've identified this. This is another way saying, okay, if I add this P protein, does it bind the bait? And this cartoon, there it is bounding. You wash there with some green protein, you get rid of that, you elute, run it on gel, here it is. And the control with no bait, there is no protein bound. Okay, so it's just another way to do it. And I think I have some arrows there. This is another company sells this little kit. It's a mobilized fusion protein here in the agarose beads. Here is the bait protein in solution. There they are finding each other. You apply them to this filter. There's a filter that is a porous filter, and it keeps proteins, a big complex, out, but regular proteins can go through. And so you wash it with a buffer, and the prey protein is immobilized here and it's bound. Then you wash away the prey. So you get rid of non-specific, and you do a centrifugation that actually pushes, and you can't tell, but this is liquid right here with your protein. You wash it down and you get rid of the non-specific. So non-specific are gone. And then what's left bound is specific bait and its prey, and then you add high salt, low pH, etc. Some molecule to break this bond. So something to break that bond to get an elution happening. And then you take what comes down here. So here's the bait by itself. Okay, so if I see two bands, which one's the bait? Well, if you do bait by itself, that's the bait. And then here's the new protein, the prey. And the control, you don't see the bait or the prey. So this is specific here. Okay, I still might do a GST. I don't quite know what they're calling an agross control there. Okay, so there are specific examples out there in the literature of using GST pull downs. So I think in plant cells there is a protein, and I don't know how they got this name, but it's PTD1. And then there is a receptor that's also a kinase. It's called a receptor kinase, EMS1 receptor kinase. Signaling of cell fate, that means cells, if they receive the signal, can turn into one type of cell. This is kind of in developmental biology, or another type of cell. Okay, sometimes our muscles got the right signal to become muscle cells or kidney cells. So this is just testing these two, see if they interact. So they did two hybrid, and there was a suggestion they mapped a region of this receptor that might handshake. And then we asked, okay, can we confirm what was seen in yeast 2 hybrid? Is there a specific domain on this EMS receptor here? And this is the paper, if you want to go read it, Signaling of Cell Fate. And it looks like in plants and animals, reproduction requires cells to become specific types of cells. So EMS1 is a leucine-rich repeat receptor-like kinase. Oh my goodness. So you might, in biology, we like to give the same protein multiple names. So EMS1 is known as EXS, and it's in a family that are known as leucine-rich receptor-like kinase receptors. Oh my goodness, and here's that abbreviation. And they have this PD1 small protein that regulate somatic non-sex cells and sex cell fate in this plant, Arabidopsis. So they express that in these cells, and it causes differentiation. And it causes an abnormal issue if they put this protein in these cells in this plant. However, they found in their experiment that if you put that protein in, it needs a functional EMS1 to do its job. So yeast 2 hybrid pull down and coabunal precipitations were further demonstrated that the TPD1 interacts with it both in the cells, in vivo, and in vitro. And it induces the receptor to become phosphorylated, that becomes active. And it's a ligand that becomes active and then changes the cell fate. So that's what this abstract is discussing. So what is some of the results? This is a GST pull down, and it's 4 different pull down reactions. So each lane here, And notice lanes 2 and 4, there's no protein pulled down. But here is the control, GST by itself with no bait and no other proteins. And you do a Western with a tagged PD1. So we do a Western, have an antibody to the high on a Western. So this is a Western blot here. And this is the pull down procedure. And they took all the reactions, pulled it down, ran it on a Western a STS page, and then Western blotted it, and then used an antibody to detect. So the control GST by itself, in the presence of the tagged PDB1. In fact, in every pull down, they added this protein. So plus means added, minus it was not added. So if you just add GST with no bait, does it pull down this complex? No. So GST shows that whatever we see is going to be specific. The next they use and mix a GST tag EMS, the complete protein, okay, with a binding domain they've named TIR within a TIR binding domain. So it's the complete protein, here it is, with that. So if they add the complete protein and add the tagged high protein, pulled down, take what pulled down in the GST resin, released it, did a Western, yes, they found it. So the complete protein and this bind together. That's what we conclude. If we break that protein up and just use the domain only that is predicted, that's right here, and that's right here, with the protein, what happens? Sure enough, just that domain will also pull down that. What about the rest of the protein? So they took the whole protein and cut the TIR domain out. Whole protein minus TAR right here, and then added it nothing. So it's very clear TIR domain is required for this interaction here. And they talk about the amount of protein. This is another paper, and they're looking at the domain of a nuclear factor in T cells, NF AT, and an enzyme calcineurin, which is a calcium-dependent serine-threonine phosphatase. So calcineurin will remove phosphates from specific proteins at the serine and threonine residues. So do this nuclear factor bind to calcineurin? Okay. So this is showing 18 different constructs. So in molecular, we're going to talk more about gene constructs. But here's GST tagged with the NFAT protein. And you already see two boxes here, CNBPA and B. Well, long story short, there's actually two areas for binding to calcineurin, not just one. So when we've been talking about pull downs and domains that bind in a pull down, we just kind of been talking about a singular. It turns out that some proteins can have multiple domains that bind a protein. So this is amino acid 2 to 414. So this is almost the complete protein, missing the first methionine, and they added the enzyme glutathione transferase to that. So it's a hybrid protein. And then they did a construct 2 to 370, and you see they chewed it back here. Then 118 to 370, notice they chewed up here and chewed back here. So see how they're chewing apart this protein? And notice they chewed so far that they chewed through the first box and chewed through the second box. And so what happens is they measure the pull down on the GST resin, and we get super binding with the whole protein, some binding here with box 1, right here, C1 plus, no binding with this region because you don't have box 1 or 2. And then you restore binding. Notice they're chewing, but adding this back. And then about right here, they chew into the box and destroy the binding domain. Nothing. They just find the binding domain and it works. Kind of like we did the TST experiment, the TIR domain. And then they chewed through that and nothing. Okay. They took this region that is from the box this way, nothing. They took that region and they found. So they were probably very surprised here because most likely they were thinking, oh, we got one. Now we have evidence of a second binding motif or domain. And look, they chew it and they chewed through it. I don't know why that keeps coming up. But anyway, there it is, nothing. Then they isolate the complete box. And then here is the pull down Western here, right here, and showing and testing the different constructs. And then they're just showing the amino acids in these two boxes. So bioinformatically, you could run these two sets of amino acids and find, are there other proteins that bind to these boxes, or there are other proteins that have these boxes that might bind to calcineurin. And so the idea is these two boxes synergize together to really give double the affinity, right? But just one box, you have some binding, but only when you have both boxes will you have super affinity, okay? So that's a cartoon showing experimental interactions. Here's just the way they may have done that to run those construct lanes there. And then there is pull down assays, and you can read that, and we've talked about that. And this is just looking at pull down and hiss and GST, and they're claiming that maybe polyhiss is more sensitive, it may be more junky. This is just some website, and this shows the different lanes 1 to 5, and then 6 to 11 if you want to go into this experiment further. So I think I'm done. Our last technique is called a far western. So don't go too far, come back and we'll go through the far western technique.